Hey, Bio30s, and welcome to our third and final discussion on Mendelian genetics and what he brought to the table. Today, I'm going to be discussing actually the shortest notes section in all of Bio30. We're only going to cover one page in the notes today. That's it. We're going to be discussing what are called dihybrid crosses. Now, you might remember when I talked about Gregor Mendel, he did his experiments with the pea plants. He took 7 traits. Each of those traits had 2 variations of the trait. He discovered that they were controlled by a dominant and a recessive allele. He called them factors, but we know they're alleles. We're going to go back to that. We're not going to be dealing with this today. We're not going to be dealing with any kind of codominance or incomplete dominance. When we're dealing with dihybrid crosses, at least for this course, these dihybrid crosses are basically going to be an expansion of what we discussed in the first video, which were our simple, complete dominance monohybrid crosses. Now, I mentioned that Mendel did these experiments with the pea plants. There were 7 traits in total. And sometimes what he would do is he would look at the combinations of a single trait, but sometimes he would breed and look at the numbers for potentially 2 traits at the same time. So in the example on the page in front of you, they're looking at whether we're looking at the pea pods in this example. So he's looking, or when he did it, one of the experiments he did was he would look at whether the pea pods were yellow or green, and at the same time whether they were wrinkled or round. So he was looking at essentially 2 sets of alleles at the same time. So in the case of the round allele, so there's the round allele first. There's I guess it wouldn't be round. It would be in the case of the allele that controls the shape. Let's call it the shape instead. On this page, they're using the designation of R. And there's R and little r. And capital R in this case is going to be, let me just make sure I got my things right here. They are, round is dominant. So round is dominant, and wrinkled in this case is recessive. Okay, so I've got my 2 alleles. I've got capital R for my round shape. I've got little r for my wrinkled shape. And at the same time, I also have an allele that's representing color at the same time. And that one was designated as Y, at least in this example. Yellow is dominant there, and green is recessive. Again, using a legend to help me write down all my alleles so I know what my designations are for all the alleles. You should be doing legends for your Punnett squares. It just makes it easier if you have someone assessing them or marking them to indicate what each of the letters represents. Don't assume that I know what your letters are. You should be indicating at all times what the letters are. Okay, so what in the example here is he basically bred initially, he bred purebred plants that were dominant versus recessive. So remember before when he had his dominant, you had 2 letters that represented the 1 allele, but now we're dealing with 2 alleles. So in the first breeding, it was represented like this. The first generation that he bred would have looked like that. He would have had one that was homozygous dominant for the round shape. It was always producing round offspring and was always producing yellow. So this was a plant that was always producing round yellow pea pods, always. So it wasn't just one factor that was determined. It was actually 2 traits that were being determined by 2 different, in this case, factors, or what we call genes or alleles. At the same time, he would have bred it with a purebred plant that was always breeding wrinkled green offspring. Well, I don't have to run a Punnett square to figure this one out, because this parent can only give capital R's, this parent can only give small r's, this one can only give me capital Y's, and this one can only give me little y's. So we pretty much know what the first generation of offspring were going to be. They were going to be heterozygous for both traits. So in this case, in my first breeding generation, I would wind up with heterozygous plants that were all going to be Phenotypically, they were all going to be round and yellow because they had one copy of the round dominant allele and they had one copy of the shape capital Y yellow allele, which was going to dominate over the wrinkled and the green allele respectively. They're there, but they're just not being represented because this is a complete dominant scenario. Again, this isn't codominance or incomplete dominance. So if I take these offspring, now I want to run a Punnett square for them. What if I took that offspring and bred it with another one of the same type of offspring? What would my ratios be? This is sort of going back to the very first thing we talked about in genetics, where he bred for purple versus white flowers. He had all the flowers in the first generation were heterozygous. They would have one purple allele and one white allele, and then he bred them in the next generation. Two heterozygous plants got bred together, and then he wound up with that classic 1 to 2 to 1 genotype ratio, one homozygous purple, two heterozygous with purple and white alleles, and then the one homozygous white. Those were my genotype ratios. And then my phenotype ratios were 3 of the plants exhibited a purple color, and only one of them represented a white color. Well, that ratio isn't going to work when I've got 2 alleles. You can see there's a bigger problem at hand here. I can't run a 4-square Punnett square if I've got 2 sets of alleles that I'm looking at the same time. I have to quadruple the size of my Punnett square. My Punnett square now becomes a 4 by 4 Punnett square with 16 squares in total. Now, how does it work? Well, here's where I need to do some erasing just so you can kind of see what's going on. I'm going to erase my legend off the top. I'm hoping you remember that. If you have to look at the legend, just rewind the video back to see the legend at this point. So what I've got right now is I want to see what my combination is going to be if I've got this is my breeding combination. I've got 2 heterozygous plants that are heterozygous for both my round pea pod shape and my for the shape of the pea pod, and they're heterozygous for the color of the pea pod. But in this case, they're all going to be phenotypically, they're all still going to be round yellow, but they do carry the wrinkled and the green alleles. So how do I run this into my Punnett square? This is what you do. These 4 alleles, the combination of those 4 alleles are going to get placed on the one side of my Punnett square like normal. The combination of these alleles is going to get put down on this side of the Punnett square like normal. The problem is now there isn't just 2 combinations. There's actually 4 combinations. So here's what I do. I have to represent at least one of the alleles always has to be R, and one of the alleles always has to be the Y allele. So here's what I'm going to do. Combination number 1 is going to be this one right there. There's combo number 1. Capital R, capital Y. Because I've got that allele could mix with that allele in the gamete when the gamete is formed. It could be 2 dominant alleles that wind up going into the allele, potentially. Into the gamete. Combination number 2 is going to be little r with capital Y. So I could have a combination where I've got a recessive allele for shape combining with a dominant allele for color. That could happen too. Combination number 3 is going to be capital R combining with little y. So in this case, I could see the dominant allele for shape could combine with the recessive allele for color. And the final combination is going to be little r combines with little y, and that gives me my 4th combination. So that's how I create the one side of my Punnett square. Now, the good news is my other parent has the exact same combination of alleles as the first parent. So in this case, this side of my Punnett square is going to have the same designations as I have on this side. So I'm just going to kind of go in order like I did before. Here's combo number 1, capital R, capital Y. I have a little r capital Y. I've got capital R little y, and then I've got little r little y. So now I've got my Punnett square all formulated up. Now I just have to kind of connect the dots. So here I've got two capital R's coming together with two capital Y's. So that's going to be homozygous dominant for all the traits. This one here is going to be little r with a capital R and a capital Y with a capital Y. This one's going to be capital R, little y, capital Y. And you'll notice what I'm doing here is I'm keeping this consistent to save myself a little bit of heartache and sorrow. What I'm doing is this Y always comes before that Y. That's just something that I do, but I like to keep it consistent so that I'm not flopping my alleles. I know this allele comes from this side and my second allele comes from the top. You could do it the other way if you want, but for me, that's just the way I keep my alleles, so I know that I'm not making a mistake when I'm combining my alleles into one of these really big 16 square Punnett squares. Again, I'm going to have little r here with a capital R on top, little y here combining with a capital Y on the top. And then I basically go through and I run all of my other letters. So I'm going to do this very quickly now. Capital R, little y, capital Y, capital Y. I'm going to go little r, capital Y, capital Y. I've got capital R, little y, capital Y. I've got little r, little y, capital Y. On the top, capital R, capital Y, little y, little r, capital Y, little y. Big R, I'm just going to keep saying it so I don't. Capital R, little y, little y. Little r, capital R, little y. And then I've got capital R, capital Y, little y. Little r, capital Y, little y. Capital R, little y, little y. And then I've got little r, little y, little y. There you go. There we go. So now that's all of my combinations in the Punnett square. Now I need to kind of figure out what my genome and my phenotype ratios are. Okay, well, let's do that. So when I look at this, I'm now going to look to see if I've got, remember this is complete dominance now, so I'm just looking for anything that's dominant. I'm looking, there's going to be 4 combinations here. There's only going to be 4 combinations that I can have. I can have a pea plant that's born that has both the dominant traits. I could have a pea plant that's born with the dominant of the first trait but not the second. I could have a pea plant that's born which is dominant for the second trait but not the first one. And then I could have a pea pod or a pea plant that's born with recessive traits for both the first and the second trait. That's what my ratio is going to be. So this ratio is going to be something to something to something to something. It's going to be a 4-letter, and it's all going to be based on a number of 16. Because I got a 16 Punnett square here. So what I'm looking for here is I'm going to go with the dominant dominance first. I have a feeling the dominant dominants are going to be the most common one out there because it's a heterozygous mix. So I got dominant here. Yeah, that's going to be the dominant. This is dominant. Even though it's heterozygous, it's still dominant for both. This is dominant for both. That's dominant for both. I'm just looking for at least one capital R or one capital Y, and then I know it's dominant for both traits because it's complete dominance, right? That's dominant for both. That's dominant for both. That's dominant for both. Dominant for both. Dominant for both. And that's it. Okay. I'm going to use different colored check marks here just so you can see all the different colors. Let's do recessive for both traits next. We'll do that one last. Let's do dominant for R, but recessive for Y. Let's see how many of those we've got. I'm going to use red for that one. I'm going to use green for that one. Green. Dominant for R and recessive for Y. All the check mark ones I can skip over. I've got dominant for R, recessive for Y because that's homozygous recessive. There's a homozygous recessive Y as well. And I got a homozygous recessive Y over there. So I got 3 that are dominant for R but recessive for Y. Now let's do how many are going to be recessive for R but dominant for Y. I'm going to use blue for that one. I've got 1, 2, 3. We're good. And then finally, which one or how many are going to be recessive for both? There's only one left. When I'm done that, if I've got two heterozygous parents in a dihybrid cross, there is a standard ratio. Just like there's a standard ratio in a monohybrid cross when you have two heterozygous parents. You've got a 1 to 2 to 1 genotype ratio, and you've got a 3 to 1 ratio for the dominant trait versus the recessive trait. In this case, the genotype ratios, we're not dealing with a genotype ratio because there's way too many genotypes here to be figuring out. We're trying to figure out a phenotype ratio only. It's just easy. It's just easier that way. There's plenty of different genotypes at play here. I just want to know what the phenotypes are. Well, the phenotype ratio, remember, it's 3 to 1 in a monohybrid cross if I've got two heterozygous parents. In this case, I've got 1, 2, 3, 4, 5, 6, 7, 8, 9 of them that are dominant for both traits. So there's going to be 9 that are going to be round and yellow. I'm going to have 3 that are going to be dominant for round, but recessive for the color. So I'm going to have 3 that are round green. Those are going to be my green check marks here. Good choice, I guess. Then I'm going to have yellow wrinkled. I'm going to be recessive for the R allele, but dominant for the Y allele. So I'm going to have wrinkled shape recessive, yellow color dominant. So there's going to be 3 of those as well, because there's 3 blue check marks. And then my remaining offspring are going to be recessive for both, which means they're going to be wrinkled and green. 1. There's my phenotype ratio. It's 9 to 3 to 3 to 1. And guess what? You could have read the slide, and there it is right there, 9 to 3 to 3 to 1. That's basically how you do it. I'm going to give you one more. Because this is nice and easy if we always have the heterozygous mix. I'm even going to give you the genotypes of the parents. We're going to use the same allele. I want you to run a 16-square Punnett square for me. I want you to figure out the phenotype ratios of the offspring, all right? So this is going to be a bit of a non-standard one. I want you to figure out the phenotype ratios, and this is the same allele, so it's going to be R is going to be my shape allele, round is dominant to wrinkled, Y is going to be my color allele, capital Y is going to be dominant to little y, and capital Y is yellow, little y is green. Same configuration that's on this slide right here. Okay, here are the parents I want you to mix together. Yeah, let's do that one. I've got 2 parents that are both, the phenotypes of them are both round and yellow. But one of them is homozygous for the round, is homozygous round, heterozygous yellow. The other one is heterozygous round and heterozygous yellow. Run them into a 16-square Punnett square, see what you get for your ratios. Go. All right, so here's what we're going to do. Combinations. This one up here is going to be little r, capital Y, little r, little y. I've got capital R, capital Y. I've got little r, capital. No, I've already got that one. Sorry. 1 and 2, and it's going to be capital R, little y. This one is going to be actually an easier Punnett square for me. I'm going to show you a cheat here because the combinations are going to be the same here for a couple of them. I'm going to have capital R, capital Y, capital R, little y, and then I'm going to have capital R, capital Y, capital R, little y. You'll notice that these are the same as these. So to save myself time, I'm just going to box out that section of my Punnett square. The reason why is This is going to give me the same information as this. So when I get my ratios here, I'm just going to double it if need be. Obviously, you want to take the smallest ratio possible. But in this case, the ratio is technically out of 8, not out of 16, because these combination of alleles are the same combination of alleles here. Whatever I write down in these two columns is going to be identical to these two columns because that's equal to that and that's equal to that. So in some cases, you don't have to write down all 16 of your squares. If you get a homozygous combination in a dihybrid cross, that's usually a good thing because that actually shortens the amount of work you have to do. The only time you really have to do all 16 squares is if you've got a dihybrid cross that's heterozygous, and then you don't even have to do the work because in theory you should know the ratio. It's going to be 9 to 3 to 3 to 1 if you get to know that really well. Hopefully, I know that when I see two heterozygous dihybrids, then I know, yeah, it's going to be 9 to 3 to 3 to 1. I know it's going to be 9 both dominant. 3 are going to be dominant for one, recessive for the other. Flip the other 3, and then the last one is recessive for both. That's a universal truth that'll save you a lot of time on exams or quizzes. But in this case, I got a ratio of 8. So let's run it through. I've got little r, capital Y, little r, capital Y, capital Y, little r, capital R, capital Y, little y. I've got little capital, I've got little capital, little. I've got capital, capital, little, capital, little, capital, and I've got capital, little. Okay, so now I got to figure out my genotypes. So I'm looking for the ones that are all capital on both sides, 1 capital on both sides, 2 capital on both sides, capital on both sides, capital on both sides, capital on both sides, capital on both sides. So I've got 6 out of my 8 that are going to be dominant for both traits. They're going to be round yellow, and then I've got essentially only 2 that are going to be round and that are going to be green. So I've got essentially a 6 to 2 ratio, which I don't have to expand back out to 16 ratios. We always want to shrink them down. In this case, that shrinks down to 3 to 1. So the ratio would be 3 to 1, and then I should indicate what that is. This would be 3 would be yellow, would be round yellow, and then my 1 would be it's going to be round green. There's no wrinkled offspring. And the reason why there's no wrinkled offspring, it's obvious. There's a homozygous parent for the shape color, so there's no possibility of getting an offspring that's going to have a wrinkled shape because you need at least one recessive allele from each parent in order to make that happen. So there you go. That's basically how you do dihybrid crosses. It's one single slide. It was a fairly long video for one slide, but again, hopefully that makes sense now. And that's about as complicated as your Punnett squares are going to get. We are going to shift gears after this. Starting next day, we're going to start talking about Thomas Hunt Morgan and the experiments he did. I mentioned him earlier in the course, his experiments with fruit flies when we discussed the discovery of DNA. I briefly mentioned him, but we're going to discuss him a little bit more about what he learned and how he sort of extended and took what Mendel did and ran with it early in the 20th century and really kind of opened the door on what genetics was. But for now, what I'd like you to do is work on a booklet entitled Dihybrid Crosses. That's the one you want to complete for next day. And at that point, we'll get started on a little thing called gene mapping. That'll be what we'll be discussing next day. Thanks for tuning in, and hopefully you have yourselves a great day. We will see you real soon.